Hello friends, a very warm welcome to Ashish Biology. Today we will discuss about exceptions to Mendelism, and this will be the second part of the chapter Principle of Inheritance and Variation. If you are new to this channel, please subscribe the channel and press the bell icon for getting more updates. And if you have not seen the first part of the chapter, I will mention the link in the description box so that you can watch the video. Then if you are I am Ashish Kumar Das, a student of MSc Animal Biology, University of Hyderabad. So what will we learn today? We will learn what is test cross and what is its purpose. That is, for what purpose this is used. Then we will see what is incomplete dominance. We will also see how ABO blood grouping is an excellent example of multiple allelism and codominance. We will also see what is multiple allelism, what is codominance. Then we will see what is pleiotropy and examples of pleiotropy. Then what are complementary genes and how they show their action. We will also learn. So first of all, what is test cross? So as the name suggests, test cross means it must be testing something. So what does it test? So it is a cross to determine the genotype of the individual with the dominant phenotype. So here we will determine the genotype of the individual. So genotype of which individual? Genotype of the individual with the dominant phenotype. So the dominant phenotype example, that is the genotype, can be homozygous dominant. For example, if you are considering the plant height, it will be TT, so it will be homozygous dominant. It can be heterozygous like capital T and small t. So we will see how this is used to determine. So a plant with the dominant phenotype is crossed with an individual homozygous recessive for the trait. So what happens if we are determining that is the genotype of the dominant phenotype? So what happens? The plant is crossed with an individual homozygous recessive. For example, if you are going to determine that genotype, that you have to cross that plant with a homozygous recessive for the trait. Since the cross is used to determine the genotype of an individual, this is called as test cross. So here you can see monohybrid test cross. So you are having a dominant character flower, so here dominant phenotype, and you don't know the genotype, so genotype unknown. So this is the violet flower, you can see. So it can be having two different genotype, that can be capital W and capital W small w. So if you are taking this, you are crossing with the homozygous recessive, that is white flower, both cases homozygous recessive, and what you are getting here, there is dominant allele is there, both are dominant allele, that is capital W. You are getting all the flowers violet because this is the capital W, and here if you are taking capital W small w is there, so you are getting half of the flowers are violet and another half are white. So you don't know the genotype of the dominant phenotype, that is the plant showing the dominant phenotype. So how you will know? So you will know by using test cross. So here you have to take homozygous recessive. So here these two genotype you don't know, you are getting these results, then you can infer that the genotype can be capital W. So you are getting this type of that is the result, you will confer that it will be capital W or homozygous dominant. If you are getting that is half of them that is the dominant phenotype and another half is the recessive phenotype, we will see you are getting that is you are having the genotype, that is the plant is having the genotype, that is heterozygous genotype. So this case the flower color is taken as the trait or character. Now dihybrid test cross. So as you know, dihybrid means it is involving two character. So here you can see homozygous recessive we have taken, that is the small r small y, that is these are the gametes formed by small r and small y. These are the four gametes we have got from this, and here you have taken this, that is the capital R small y, that is capital Y, capital R small y you have taken, and you don't know which one is actually the genotype. So if you are doing the test cross, we will get the result like this, the dihybrid test cross ratio, and from that you can determine what is the ratios and what that is the genotype and phenotype. So here the plant that is taken is that is from these four gametes are from the plant, that is capital R small r, capital Y and small y. So back cross, what is back cross? Backcross is the process of crossing a hybrid with one of its parents or individual genetically similar to its parents. So you have to cross a hybrid, that is the heterozygous mostly, crossing it with a parents, one of its parent or individual genetically similar to its parents. So here you can see parent crossed, that is A and R, recurrent parent you can see R, then you are getting B, again this is crossed with R, so this is called recurrent parent because it is used again and again. So this is test 2, test 1, test 2 you can see. So T generation, T1, T2, T3 you can see like this. So F1 you can see, so the R is crossed with the, that is the offspring you are getting again and again. So this is done in order to obtain a offspring with a genetic identity which is much closer to that of parent. For example, a parent is having very superior quality. For example, in case of plants product, it is having better yield. So you have to retain that genotype. So it is used to get the offspring with greater genetic identity to the parent. So it will serve our purpose. So this is used in the horticulture, animal breeding and production of gene knockout organisms. So gene knockout organisms means you have to eliminate some effect of gene, you have to add some effect of new gene in order to increase the productivity. So these are used in horticulture, animal breeding and production of gene knockout organisms. Now incomplete dominance. So we have seen what is dominance and incomplete, that is the dominance and recessive character. But here incomplete dominance means the dominance character, that is the dominant, is not complete. So not complete means the one of the allele is not able to suppress the effect of another allele to, that is the 200% extent you can see. So here you can see this is an example of the Antirrhinum majus, that is snapdragon plant, and it is also called as dog flower plant. So this is an example of incomplete dominant. So the flower color in the Antirrhinum majus is a example of incomplete dominant. So here you can see a red flower, that is capital R, and a white flower, so smaller. So from here we can infer that the capital R, that is the red, is the dominant and the white is the recessive. So now you are getting a pink flower. So why you are getting pink? Because the red color, that is the capital R gene, is not able to suppress the small r gene completely. That's why you are getting a pink flower, that is capital R and small r. Now the gametes you can see, the gametes here you can see, that is the capital R and small r, here capital R and small r. So here what happens, if it is both capital R, you are getting red. One is capital R, another is small r, you are getting pink, and another other, that is smaller, you are getting white. So what happens, the phenotype ratio is 1:2:1 and the genotype ratio is also 1:2:1. So this is a case of incomplete dominance. So this is a deviation from the what Mendel has predicted, that is dominance and recessive, that is 3:1 ratio and 1:2:1 ratio. So this is a deviation from the Mendelism. Now multiple allelism. So as the name suggests, multiple allelism means more than two allele controlling, that is present in a single gene, and we will also see co-dominance. So what happens, here the classical example is the blood grouping, that is ABO blood grouping. So gene for blood grouping exists in three allele, that is capital I, that is I capital A, I capital B and small I. So any individual carries two of these three allele. So as you know, the human beings are diploid organism, and what happens, one of the set of the chromosome we are inheriting from our father and another set of chromosome we are inheriting from our mother. So any individual can carry two allele of, that is two allele of the three allele, that is out of the three allele a organism is having two allele. So IA produces glycoprotein A, that is I capital A produces glycoprotein A and another antigen A. So this is also called as antigen A, and this is present on surface of RBC. I have already uploaded a video on blood grouping. You can see that video. I will mention the link in the description box, and in there I have completely described what is antigen, what is antibody, etc. Then IB, that is I capital B, produces glycoprotein B called as antigen B, and that is always the antigen is present on the surface of RBC. So IA and IB, that is I capital A and I capital B, that is capital IA, capital IB, you can say. So they are co-dominant, that is they are dominant on each other, but both are dominant over I. So what happens, co-dominance means if I capital A and I capital B is present, what happens, none of them can suppress the effect of another. So both will be expressed. That's why they are codominant, and they both are dominant over small I. So this, from this table you can see, so allele from parent 1 is there, allele from parent 2 is there. So let parent 1 be mother, and parent 2 be father. So here you will get I capital A, so it will be that is capital IA, blood group A, capital IA, capital IB, blood group B, capital IA, small I, blood group A. So as you can see, if IA is present, then most of the cases it is becoming blood group A, and if both are present, IA and IB, it will become AB, because both are codominant. So they will express both the antigen on their cell surface. Then you can see here also IA, IB, both cases are same. This is interchange the position here, IA, IB, and IB, IA is interchange. Then you can see capital IB, it will be blood group B. Then capital IB and small I, it will be blood group B, and capital I, it will be blood group O. So here you can see the different blood of the offspring. Now come to pleiotropy. So single gene product may produce more than one effect. So in case of multiple allelism, we have seen there are more than two allele and they are controlling a single character. But here what happens, a single gene product may produce more than one product. So it will be more than one effect. So what happens, sickle cell anemia, this is an example of pleiotropy. So mutated beta chain, as you know, in case of sickle cell anemia, what happens, the 6th position of beta globule chain is replaced, that is glutamic acid is replaced by valine. So what happens, it causes blood clots, high blood pressure, and loss of vision. Sickle cell anemia, that is the change in the shape of the RBCs, that is from normal spherical shape it will become sickle shape. So it is also associated with the blood clots, high blood pressure, and loss of vision. So this is an example of pleiotropy, because the single gene, that is the beta gene, is controlling, that is HBB gene, is controlling the other, that is the blood clots, high blood pressure, and they are responsible for this. Then seed shape and size, that is the starch content. So here this is an example, the parent, this is also seen on plant. Then small b, you can see big round and small wrinkled. So here what happens, the capital B gene is controlling both the round shape and the size. So B is controlling shape and size. So similarly you can see here, so if it is capital B, it will be big and round. Capital B and small b, you are getting intermediate size and round. So capital B, small b is intermediate size, and small b is small and wrinkled size. So here what happens, this is a deviation from the Mendel law of dominance. How it is deviation? Because what happens, the capital B is there, but it is not showing, that is not able to show its dominance completely. That's why this is a deviation. And this is also an example of pleiotropy, because the single gene is also controlling the seed shape, that is the, you can say the seed shape means round or wrinkled, and it is also controlling the size, that is big or small. That's this is an example of pleiotropy. And again this is a deviation from Mendelian's law, because the capital B gene is not completely dominant over the small b. That's why this is an example of, that is example of the incomplete dominance, you can say. And this is also an example of pleiotropy, because the capital B is controlling, or both small b and capital B is controlling both the shape, that is the round or wrinkled, and the size, that is big or small. So here you can see the diagram, the beta globin gene mutation in sickle cell anemia. This is also causing blindness, liver failure, and heart attack, that is maybe strokes or blood coagulation, that is blood clotting in the hearts. So various problems. Now come to complementary genes. So complementary genes means, as the name suggests, they complement the function of one another. So when two genes, that is non-allelic, that is they are not having same allele, they are having different allele, two genes complement the effect of each other. That means they will enhance their effect, each other effect, when they are present together. If they are present single, then they will be doing any different action, and if they are present in combination, they will be doing any different action. So here you can see flower colony, the Lathyrus odoratus flower, that is the white variety, that is capital C, small p. Here you can see small c and capital P. So both are white here. So if you are having capital C and capital P, then only it will be purple. So when they are crossed, so obviously the dominant will be there, so the dominant character will be inherited from in the, that is, will be obtained in the F1 hybrid. So here you can see the purple is there. So when you are selfing the F1 hybrid self fertilization, you are getting 4 types of gametes here. And again you are drawing the checkerboard and the, you can say, Punnett square. This is developed by Reginald C. Punnett. So you are getting the Punnett square. So here you can see only this combination, that is the capital C and capital P combination, will be purple. So there are 9 purple, these are the purples, and others will be the, that is all, if only capital C is present or only capital P is present, then they will be white. So here the ratio is 9:7 ratio. So this is an example of non-allelic interaction, you can say, and there are a number of non-allelic interaction like duplicate gene, supplementary genes, epistasis, etc. But at your level only 1 or 2 is knowing, that is not in your syllabus, much more is not in your syllabus. That's why this is sufficient. I thought this is sufficient for you. This is the end of the video. Thank you for watching.